Good evening, everybody, and welcome to the 9th edition of the Oracles of Eye Innovation webinar. I'm Chris Morrill, one of the co-founders, along with Eileen Kilich and Dr. Michael Moroccan, who cannot join tonight. With us tonight, going from my right and counterclockwise, Dr. Sufi Tanari, who will be moderating tonight's panel discussion, Professor John Marshall from the UK, Dr. Eileen Kilich in Istanbul, Professor Gerd Alfarth, who is in Heidelberg, Germany, and Professor Rudy Nuijts, who's joining us from Maastricht, the Netherlands. Good evening, everyone, and thank you for joining us. Tonight's topic is we're going to be having a discussion regarding the developments around artificial corneal technology that we're starting to see come into clinical study and hopefully into the market in the not too distant future. And with that, I'm going to turn it over to Dr. Tanari to share some slides and make some initial remarks before we get into the discussion.
Thank you, Chris. So without further ado, I would like to talk about why we need better cornea graft materials. Materials that we put into the cornea or on the cornea or underneath the cornea, because in the past, we had not so pleasant experiences. Here you can see an AcuFocus implant, although it has been removed. So removable is not reversible, that we have learned the hard way. Another entity of complications is here shown in a keratoconus patient. He had a clear cornea with Fleischering and Vogt-Stier, and we recommended keratoconus cross-linking to him. However, he underwent femto-assisted implantation of 2 intact segments, and shortly thereafter they removed the superior segment because of new vessels in 3 quadrants, and they left 1 intact behind, which later then we removed. You can see the new vessels, you can see the leukocytes, and this is another problem with these PMMA material in addition to the penetration or extrusion, which has been widely published. So to conclude, I have also 2 cases of flat melting over corneal inlays for hyperopic correction. The first one is a female patient with +8.5, and the second one a male patient with +6.75 diopters of hyperopia, which we couldn't correct with the excimer laser. So this hydrogel implant is put underneath a LASIK flap. Orb scan and keratometry were unremarkable, and the post-operative recovery was unremarkable, and the findings were stable for several years. However, at 9 years, visual acuity dropped to 0.3, and you can appreciate the flap melting in the OCT. And the inlay was removed immediately. Fortunately, after 3 years after explantation, the visual acuity recovered to 1.0. So here you can see 8 years post-implantation, a clear cornea, clear implant. After 9 years, we have this melting. 1 year post-explantation, and 1 year after explantation, the cornea was almost recovered. The second patient was okay until up to 9 years. Then he noticed a sudden loss of vision, and we couldn't convince him to have his implant removed. He returned only after 2 years, and we showed him the deformation in the OCT and the melting in front of the inlay, and then he agreed to have the inlay removed. And fortunately enough, also his cornea recovered somehow, and he didn't need a corneal grafting. So in conclusion, these cases illustrate that potentially severe biocompatibility issues and other issues may occur after even several uneventful years. And that's why I think we need better grafts or better graft materials. And with this, I would thank you for your attention, and would like to open the discussion. So maybe Professor Aufert can tell us about his experiences with EndoArt.
Yeah, maybe before I do that, I can also comment on the inlays, because we were talking before we started this session. We have different ideas here. The one thing would be these functional implants that should more or less correct optical errors. Yeah, like the camera inlay, like the raindrop, for example, also, or the presbyopia inlay. And then we're talking about, and Rudy is also joining us for this, about replacement of tissue like artificial corneas, which is a different area. Maybe we pick up what you just said, that after first almost euphoric adoption of this technology, because it was easy to implant, and looked like for refractive surgeon as a good presbyopia solution. We all know colleagues who were enthusiastic for like 1 or 2 years, and then suddenly they changed to become the biggest enemies of this technology. And we can see that the cornea is a very specific organ. And I would say in general that the first 2/3 of the cornea are no good place for any implant. We can debate about the fact that the deeper you go, let's go 250, 300 micron, that we are in a more quiet zone, which may be interesting for a very, very biocompatible hydrophilic implant, and that on the long run, we may actually recover that area. But at the moment, with current technology, I think that all these things were too superficial, and in an area where we have a lot of cellular reaction to the implants. And I think that was what killed these technologies, so to say. And we learned the hard way that the cornea is not a forgiving tissue, because it cannot really regenerate so easily. Once you have an implant in, you will see it the next 30 years, even if you removed it, as you shown in the first picture. And I don't know, maybe John has also an idea about this, because he's very, very good in, he almost knows every little cell inside the cornea, personally, by name.
No, Gert, I just wonder why you said the superficial third. Clearly, that's where we have the sensory plexus. So there are lots of nerves there. It's the highest concentration of keratocytes per unit depth. But I think one of the problems here has been, ever since polysulfone inlays, which had a terrible history, part of the problem has been that of fluid movement in the system and support to the cells. And part of it has been biomechanics, putting in something which is very different from the native material. So I would agree with you. This needs very careful studies and much more detailed studies. And again, I would agree with you. I like the differentiation between splinting a diseased cornea and inserting a refractive correction. So I think we're in general agreement, and I just wish I did know more, actually. I don't speak to any of the cells that speak German in the cornea, by the way.
Oh, no, they don't. Oh. Maybe that was my problem.
Rudy,
Rudy,
go
do you want to
ahead.
comment?
Yeah, you know, the whole story about intracorneal inlays brings me back to one of my earlier teachers, which was Gabriel van Rij. You probably still know him, Gerd, or some of you. He was a corneal surgeon in the eye hospital in Rotterdam. And when he was young, he went together with, well, no, actually before Houdin Beekhuis, he went to Emory, where I also have been for my PhD with Henk Edelhauser. And Gabriel performed hydrogel implants in monkeys with Bernard McCarry at that time. And actually, the story did not, that was late 80s, I think. And the story is still the same. You know, it's unpredictable what's going to happen. Sometimes it works, but most of the times it doesn't work because there is encapsulation or there are biocompatibility issues or whatever. And I think the last sentence was very true. It makes a lot of a difference if you're going to perform a cosmetic refractive procedure, or you're going to do something to a patient who has a medical necessity to do something.
Yeah,
And like you showed in those two cases, you know, to have a result like that, it's of course dramatic. And I put in some corneal inlays, you know, in the beginning, camera, etc. And I had some you know, undesired results. And what happens then is usually that the company says, you know, that I have not seen before. You know, we have done this so many doctors, and I said, well, you know, I think I'm not a bad surgeon, and I think I'm not mad. But, you know, in two of the first 6 or even 3, I had a hyperopic shift. And if you listen to, yeah, you have to use more steroids. But if you listen to people on, you know, on a podium, it almost never appears. And that's the kind of, you know, discongruency that you sometimes have with these products, and also, you know, marketing, etc., and makes it sometimes also very difficult to judge the results at first hand. So that's another, you know, part of the equation. How can you fully judge what is happening there?
Okay, I agree with most of what you said, all 3 of you. However, I think there is no clear-cut distinction between refractive surgery and curative surgery, because the intakes were put in a keratoconus patient to improve his vision, which could not be improved otherwise. And maybe this is a good point to Eileen to join the discussion.
Yeah, so I just wanted to make comment. Of course, I am against the biosynthetic material, but honestly, I performed a lot of intercorneal rings, PMMA, and I believe so many patients could survive to postpone keratoplasty, This is important point, because everywhere we cannot do keratoplasty emergency. Each country is not same fast, and sometimes younger group, if there is progressive keratoplasty. In the past, when I perform intercorneal rings, children 10 years old, sometimes the youngest one, they cannot use contact lens. This is not permanent solution, but at least make them their life better, higher quality is a solution, even not long term. This was also only point what I would like to tell.
Thank you.
But as almost as a bridge to delaying more invasive surgery.
Yeah, but still we're talking here about more a curative approach, because you have a disease, a keratoconus, and not a normal cornea, which just has a refractive error. And you kind of delay more invasive treatment, which would be a complete perforating keratoplasty. So I still see this distinction. Yeah, if you put in a corneal ring for just the pure purpose of refractive correction, in a non-diseased eye, yeah, then I would be with you. Yeah, that this I would also, and we did that. I mean, years ago, there was also a phase where you try to correct just plain refractive error, independent from morphology and independent from a dystrophy of a cornea. And the cases you were just talking about are still, we have a disease, we delay further treatment. So I think there's still these two groups and differentiation between lifestyle surgery, more or less, and medical treatment.
Yeah, from my perspective on the commercial side, having been involved in selling or marketing many of those technologies, and one in particular, there was a period where we had to go back to the users and go over within 3 months, provide 3 different post-operative steroid regimes. Because the company, I think you probably remember this, Rudy, the company, you know, there was this hyperopic shift. There was something going on. The cornea was not responding. So the answer was to change the steroid regime 3 times in 3 months. And how do you go back with a straight face to a physician and explain that, you know, and you know, I think having been involved in 4 of these artificial technologies over the years, you know, there is that definite line that, and I think we all recognize it now, that the restorative versus, you know, cosmetic, refractive surgery, it's just, there is a delineation that people don't want to cross anymore. And I think that's one of the reasons why intraocular lens surgery has become more popular. Yes, John?
But may I ask a question, Richard? This is really directed at you, Chris. Like many of the others on the panel here, we've sort of suffered with the never-ending changes and barriers to entry of regulatory affairs, as it were. Do you think, I mean, how do you think post-market monitoring is going to affect this sort of technology, where you put in, let's say, whatever the new implant is, and if it goes through what Sophie has described, I almost think that commercially that's going to be a barrier to further development of technologies in this field, because it's going to cost the companies a fortune. And I just wonder, what do you feel that this sort of intrusive regulation, which is a good idea, let's see if it continues to work, but if it's going to have a feedback to stop innovation, I'm not sure how useful it is.
Rudy, do you want to comment?
Yeah, you know, that's an interesting question. I just had a PhD defense of one of my PhD students on the long-term effects of the artisan iris claw fixated lenses of Jan Borst. And we did, probably you know that if you have not seen it, but we did studies with 10 to 15 years of follow-up. And I mean, there is, after 10, 12 years, extensive cell loss occurs suddenly because probably the niche, you know, has no resource anymore, et cetera, Now, in the opponent committee was the head of plastic surgery and the head of orthopedics of our hospital. And they said, huh? Use a new device, just put it in an eye after almost 1 year of study. You know how long it takes to use a new hip? They have to do studies for 7, well, Chris, you probably know that it more than I do, 7, 8 years actually, before they can go to market. And, you know, in plastic surgery, because of the PIP implants in France and this all kind of stuff, you know, it's also becoming very strict. So I would predict, you know, that with at least certain innovations in our field that have seen a lot of problems like intracorneal things, et cetera, that the game will change, you know, in the years to come. For intraocular lenses, it's probably different because they are in an environment where there are not a lot of cells. If you are in the back, that probably makes a difference. But for, you know, intracorneal inlay, things like that, it probably go and using their, you know, biosynthetic polymeric implants, I think it's going to be a little bit more stringent for the future.
Yeah.
I don't know what John thinks or Chris or Gunnar.
Well, interestingly, we had a client last week whose notified body came back and asked them for all of their post-market clinical follow-up data on all of their intraocular lenses, which some stretch back 20 years, and I just laughed. Because it doesn't, as we all know, it doesn't exist. However, we also know that intraocular lenses have an exceptional safety record, and have evolved and gone to, softer materials, et cetera. John, to your point about innovation.
May I put one comment there?
Yeah.
I think there was just a company that went bankrupt because of the,
Sure.
the problems in the UK, and actually the base of the company was in the Netherlands.
Yes.
And the court in the province of Gelderland declared them bankrupt. I think last week and the week before. I say last week because he knows exactly what I mean.
Yeah, but that company had manufacturing issues, and
Listen, I mean, we knew that for how long, Gert? 8 years from now, or even longer, and still. It happened again and again and again. And I pose, certain questions for the regulatory bodies, that this can continue for so long in our patients, patient safety is still first, in my view.
Yeah, I agree.
That's where I think, John, to your point, John, to your question, that's where the MDR is not such a bad thing. And getting companies to follow more closely and report more closely their adverse events, which we know occur. The calcification issue had been around for years. So if a company who's coming into the market is starting clinicals now, is doing things correctly, they're going to be in good shape, and there's not going to be any issue post CE mark, because they will have the data, they will have the plans in place. The companies that are really struggling now are the ones who've been on the market for a while, and especially for IOLs, didn't have to do this. So we're going to see fallout. There's no doubt in my mind. Somebody asked me the other day about legacy phaco equipment. And if you think about, phaco systems that have been in your hospitals for 10 years, chances are the manufacturers are not going to go back and justify collecting clinical data on those systems. So we're getting off topic, but it is definitely a new world. It definitely is.
No, the only reason I brought it up, and I don't want to hijack anything here, was I hate to see constraints on innovative research. And when the constraints are financial, coupled with restrictions on route to market, it is worrying. But I absolutely 100% agree. Efficacy is our main goal here. It must work and it must be safe.
Yeah. And at the end of the day, the path to, sorry, Baird, the path to market, getting a CE mark has not changed dramatically.
Okay.
Yeah.
Well, one thing I would like to add is, if we talk about intraocular lenses, and we look, for example, at Germany, we do like 1 million implants a year. Yeah, so with 1 million implants, even in 1 year, you can see a variety of problems fairly easy. If we talk about corneal surgery, and now we are talking about like 10,000 surgeries maybe in Germany, and then let's go down to inlays or whatever, then we're talking about a few hundred or a few thousands, whatever. So we cannot really compare that. We have much broader experience with an implant related to cataract surgery than even with plain curative corneal surgery. And in corneal surgery, we are now seeing, 40% perforating, 60% lamellar, or even higher differences, 70 versus 30%. So we diversifying even more in the cornea business. And so we have, I think, also from a regular point of view, a different story here. But saying that, I think it leads quite good to the next discussion. We still see, similar to the increase of cataract and IOL implants and surgeries, a lot of varieties in terms of corneal surgeries. Yeah, like 10 years ago, there was a perforating keratoplasty. It may be some lamellar approaches. But now we have DMEC, DSEC, DSO, all types of lamellar procedures, and so on. And now we also see artificial implants entering that busy area.
Yeah. Yeah.
I would like to pick up on that. Yes, we are seeing artificial materials, and we are also seeing allogenic materials being developed. Artificial materials for keratoconus correction, presbyopia correction, and allogenic materials for presbyopia correction, for hyperopia correction, and also to stabilize a keratoconus by putting in a disc or a ring-shaped inlay into the cornea. I think maybe Eye-Yon can comment with her experience on ring-shaped implants for keratoconus correction.
We had opportunity to perform allogenic intercorneal rings, which is described for Susan Jacobs, but we have ready materials in the package, 2 years shelf life, and ready to use tissue. And this is not fresh cornea. This product, we use this product for 60 eyes at the moment. Our follow-up maximum 2 years. And our patients are at the moment, we can discuss about refractive outcomes, but the most important message is safety. I performed a lot of intracorneal rings, PMMA rings, and I observe some complications, of course. I was very selective, I was uncomfortable, pachymetry was very important. So many different indications was not easy for PMMA. But when I perform allogenic intracorneal rings, I am feeling more comfortable because I had opportunity also allogenic hyperopic inlay and presbyopic. It's the same tissue, allogenic. I was more brave this time to perform more superficial tunnel and larger tunnel in keratoconus, because we followed hyperopic allogenic tissue 4 years. And our observation is tissue become more clear within time. This was very important experience for us, and we have more positive, very optimistic to use also for keratoconus. Of course, these are more different patients group. I expect in the future allogenic intracorneal rings will be featured for them, alternative to keratoplasty, of course. And I expect in the future more clear, and I don't expect any reaction because of our experience. We are still following. We didn't see extrusion in learning periods. We had some few extrusion. This was the learning stage, but now we improved, and we are working still about nomogram, and we have no vision loss. This is the most important. And this group is really very special. They cannot use contact lens. They all cannot use contact lens, and always we cannot perform keratoplasty within short time. So I believe this is future for keratoconus.
Thank you, Eye-Yon. Any questions?
I think it's, this comes back to the question of tissue addition, of course, in irregular corneas or in scarred corneas. I've seen a couple of cases now and abstracts also where they use SMILE technology to remove a scar from a cornea and then put in a disc to replace that from an allogenic source. I think it comes down to quality of vision, the incidence of stromal allograft rejection that you might have. How high is that? How long do you have to give steroids in these cases? And the visual quality at the interface, because all corneal surgeons know that if you do anterior lamellar keratoplasty, one of the problems is the quality of the interface and the quality of the vision.
Yeah.
But as long as you stay out of the central part with your rings, that's probably not a significant issue. The only thing that, you know, what you ideally would have, of course, is a kind of an imaging technology that would let you to prepare customized rings to add, you know, sufficient tissue in the region where it's needed and not in the other region. That's, of course, the holy grail that you would like to see there.
We are using anti-corticosteroid 1.5 months. We are using 5 times a day, 1.5 months, and we taper it. And during follow-up, sometimes we continue longer. But as you said, we are advantageous because we are away from visual axis. And if we would be central area, also we had experience about that. Personally, we did a European study, but personally I used longer corticosteroids than other centers. Also, this corticosteroid and rejection or reaction, whatever, is related with femtosecond laser, which kind of laser you are using, which company, which energy level, these all affect inflammatory reaction. We had study, we evaluated haze degree with Pentacam degree, and our observation is first 3 months, there is a degree of haze increase, and then 6 months, there is a plateau level and stability. This was our clinical study. We presented, and very soon we will publish. And longer steroids, I agree, but in keratoconus, this is advantageous to be away from visual axis, and Gerd made wonderful comments. If you are superficial, there is more opportunity to have haze and keratocyte reaction. When you are deeper, there is less opportunity, less reaction.
Well, that really, all this relates to the cells. If you activate both the epithelium and the stromal keratocytes at the same time, you get crosstalk and TGF-beta and a hazy outcome. If you hit one of those populations only, then you don't get crosstalk, and it's minimalized. So I wonder, you know, you guys are the expert in surgery, etc., and the easiest route is to delve into the corneal stroma. Now, I know that the whole onlay issue is difficult because of rejection and how you get the onlay to stick there, but that is another potential route and another area of research. And I know that in the past you did a little work on onlay technology, and I don't want to sort of introduce side issues here, but that does have some potential if you can deal with a system after an epithelial debridement and perhaps some changes in Bowman's. You know, I think there is a potential future there. So with our current technologies and our current surgical procedures, yes, we've defined a lot of the problems, but let's think a little bit laterally for the future, perhaps not tonight, and come up with some innovation and perhaps some improvements. I see you're smiling.
I think Gerd wanted to say something.
Yeah, sorry, Gerd.
Yeah. Another way to go around this issue of the cellular reaction is to use an implant which you don't put in the cornea at all. So I'm talking about the EndoArt implant, which is outside the cornea. Yeah, the EndoArt implant is a hydrophilic acrylic implant, which looks like a contact lens, a very small one. It's like 6 millimeters, 6.5 millimeter in diameter, 30 to 50 microns in thickness. Hydrophilic acrylic, same material we use also in intraocular lens, and it's placed inside to the endothelium or on the inner side. Yeah, it attaches with surface tension, And what it does, it blocks the water going into the corneal stroma. And with this, you can treat corneal edema. So it's an alternative to a DMEC procedure or a DSEC procedure. It's very, very easy to handle in terms of surgical procedure, because you don't need this delicate handling of a one cell layer tissue, which can easily be damaged. It can fairly easily be implanted. And we have now almost 3 years follow-up in Heidelberg. Or next year, we're approaching the 3-year follow-up in March, I think. And overall, around almost 30 patients in clinical studies have been implanted. This kind of implant now works. It doesn't impact the cornea in this way that it destroys cells or something. It just prevents water from going into the central cornea, and chronic corneal edema especially is going down. And we all know that not everywhere tissue is available. We all know that there are not always DMEC surgeons who are very experienced. DMEC learning is a long learning curve compared to other corneal surgeries. And the other thing is we can actually also just treat pain. Yeah, if a patient has chronic corneal edema, he has a lot of pain usually. We would rarely do a surgery and risk human tissue just for treating pain. Yeah, what we do is, of course, pacify, then we use it. But for pain, we rarely do, just if the patient is really desperate. So we're opening here 2 categories. We clear the cornea, and also we can treat other symptoms of the patient with this kind of implant.
Can I just ask, I've watched this work and I found it absolutely fascinating, but the current hypothesis and all the cell biology says that the endothelium is pumping water out of the stroma into the aqueous.
Yeah.
So I've never quite understood how this system works mechanistically.
Yeah, the thing is, you prevent the water from coming from inside into the cornea, and the cornea itself is losing the water just by evaporating it outside. Yeah,
Surface evaporation.
Yeah, it's just going out. Yeah, it's not going back. The tricky part was the sizing. If you make it too big, you can more or less dry out the cornea and it can melt. If you do like a 10 millimeter implant or whatever, cover the whole, yeah. And if it makes it too small, the whole cornea gets thick around the implant. So over the years, there were a lot of studies done in Israel by Eye-Yon and also by the doctors that work for them. They figured out that this around 6 millimeter is the optimal thing of still getting nutrition in, keeping the cornea at a certain thickness, and so on. All the corneas that we treated with this ended up somewhere between 480, 550 microns, so around normal corneal values. Yeah.
Yep.
That's very impressive. Rudy?
Yeah, you know, I think, Gerd, I think we spoke about it before, but when I was a corneal fellow in the eye hospital in Rotterdam, 93, you know, there were still the patients from Ragnar Zivojnovic who put in silicone oil. And all these anterior chambers that were filled with silicone oil were the same phenomenon. The endothelial barrier was blocked and the corneas were clear until somebody got the stupid idea to take the oil out because they suddenly became very thick. So I think the concept is very, well, it's nice and it's easy. It's about blocking the endothelial barrier. And, you know, you talked about how the cornea gets clear. 20% of the pump function is by the epithelium, the epithelium. We know that. So, you know, that's why they become even more digest than the normal thickness, I think. You have blocked the endothelial barrier and then the epithelial pumping continues. But what I also saw, I think, on the pictures is that outside of the 6 to 6.5 millimeter, the corneas become thick. So there exists still that ring also on the surface where you have microcystic edema and you can get infections and things like that, I think. So that's a little bit that worries me in the concept. I don't know if you have seen that or not, or what, or do you keep the patients on antibiotics or?
No, we don't really need to do that. I have to say that we And I have to say, we started in corneas that had like 900 to 1,000 micron thickness because they were very diseased corneas. We didn't start in let's say DMEC cases that still could see 0.5 or something. Hand movement was already a good visual acuity when we started with this. And when then at the end of the procedure and after a year, the peripheral cornea was 700 or 800, that's not so much different from a normal peripheral cornea. Yeah, the normal peripheral cornea is not 500 microns. It could be easily 600-700. So so that was not really the issue. Yeah, we were a little bit afraid that we get like a donut-shaped type of cornea. Yeah, but the sizing is kind of preventing it. If we would take 5 millimeters, we may have that. Yeah. So I'm pretty positive that in the cases that we've seen, we will not really have that. The other thing is what you said is very important. By removing by reducing the corneal edema and also reducing bullous keratopathy, the epithelial can regrow and can also regenerate, and then can fulfill better their 20% of function in terms. So that also applies to the peripheral cornea. Yeah. So that's a kind of healing effect also for the tissue here.
Okay, let me put it in a question form. Do you think EndoArt is going to replace DSEC and DMEC and obviate the need for human corneal donor tissue?
I can clearly. Oh, yeah. You want to go first or what?
I think you also should talk about the full size replacement later, the Corneat and stuff. Now, I would say that definitely we reach a state now where we can use EndoArt as a. I'm not quite sure if it will completely replace. I think that's a little bit too much you can expect from a first generation type of new technology. Yeah, it may lead to artificial technology that can replace on the long run lamellar techniques. If EndoArt is the one, I don't know, if at least it's the first type of this thing. And so I see a lot of potential in that and a lot of more development which is possible, including even refractive correction from inside. Yeah. Okay. But the thing is, we're talking about one part of the cornea. There's still 90% of the rest of the cornea, and I think Rudy also knows some solution in terms of replacing the entire cornea with artificial material. Maybe he can comment on that and how far we go there.
You know, first on commenting on your question there, it's going to replace. I don't think so actually, because I think it will be reserved for a niche market. Those cases, you know, where we think that there is a high risk of rejection, where we have tried already a couple of times, DSEC, DMEC, you know, those are excellent cases to apply this technology, I think, and in painful eyes. But, you know, DMEC is so successful. The rejection is very low, 1 to 1.5%. And most important thing, it's cheap, you know, a graft in the Netherlands, it's 1,900 euros. So if you have to make a medical device for a niche market, and you can put that with all the research on the market for 1,900 euros, be my guest. But I think that's going to be very difficult, actually.
I think 1,900 euros is not too much, I hope this Corneat is, if it's scaled, if it's produced in a larger number, then it could be cheaper than that, actually. Plus, in a human donor tissue, you always have variation in quality, which in a product with long shelf life, you don't have.
No. I think that the DMEC grafts that we get from our banks have a very high standard of quality. You know, that the procurement, the evaluation is all, you know, up to standards. The cell count is you know, on a certain level, etc., etc. So I think conceptually, at least in the Netherlands, I think you won't find a lot of corneal surgeons who would be prepared to do this on a routine basis. They will do DMEC.
I cannot. Let me put it this way: in Germany as well as in Turkey, there is a shortage of donor tissue, and tissue is imported from the US. So that's a huge problem, apart from the cost. The availability, the quality, and the cost, and variation in and short shelf life. Eye-Yon, what do you think?
In Turkey, we have a waiting list about cornea, and then we buy from abroad. This is usually insurance doesn't pay, and we cannot use this tissue. We just can use, as I know, only foreigners, not Turkish people. If it is emergency, we can find immediately, but we have waiting lists, and we cannot find immediately cornea always. Tissue, of course, iBank is working very good, very nice, but always to find cornea donation is not easy every country.
No, but I understand, that cornea donation in certain countries, because of whatever cultural, religion reasons, may be a problem. But I can only speak, for my country, and in my country, there's absolutely no shortage of tissue. The only problem from the waiting list now are our covered problems and the capacity in the academic centers to compete with all the other subspecialties for the surgical time. That's the reason for the waiting list, not the shortage of tissue. Actually, it is even worse. They have to put sometimes tissue into the garbage can because they cannot distribute it enough. And we actually, I think we export to a certain extent to Germany, as far as I know, some of the tissue also, but at least we don't have that. And I know in the States, we said it already, in the States, there is no shortage of tissue. But, I understand there is shortage in other parts of the world. But, in, let's say in Asia, in Nepal, in all these countries where there is a lot of corneal blindness, there is no market, I think, to pay a lot of money for an EndoArt device, actually. So, I mean, if you have to go in there and solve that problem, then you come into kind of a price elasticity issue.
What make you think that the EndoArt is so expensive?
Well, I would say, if you have a product that has not an enormous magnitude of scale, and you ask a company, to put everything in place, not only the research, the marketing, the quality assurance, the registry, and things like that, it's very expensive. John knows that much better. I mean, if I speak with companies, the production of that product is only a very small part of the whole stuff, but the rest around it, that takes up all the money. And Chris, I think, can probably testify to that. And so, it all comes down to, can you scale up and how much you can scale up, I think.
Yeah, and you're absolutely right, Gert. I actually like the EndoArt concept because you could end up with a graded index lens in the cornea, which would be a very interesting refractive element. And really, you think you've got problems. In the UK, with less than 2,000 ophthalmologists, we now have a cataract list going into 2023. So it's really appalling. We may be the first country in Europe that puts cataract back on the list of blind registration. I know that it's awful.
Yeah. But to Rudy's point, I mean, Rudy, you said there are times where you have to bin tissue.
Yeah.
If you have something that's sitting on the shelf,
sure,
that you can rely on, and not have to worry about throwing away some precious donated tissue, is that a certain advantage over the natural, the human tissue alternative, sorry.
I also think that on the long run, a human tissue may become even more expensive, because to have a cornea bank, and I can tell you from myself, is very expensive. Yeah, you have staff to pay. I pay around 200,000 euro a year just for a simple staff, and some other things. The regulatories to produce, sell, and use human tissue is a nightmare. We have audits. We have approval from local authorities. So having a cornea bank is quite expensive. You can commercialize that and have a network. Yeah, but then you can also dictate the price in a way. Yeah, and like an implant like the EndoArt will definitely much less than you pay for a human tissue. And you can just have it like a lens in a stock, so to say, or have like 10 of those in your stock in the clinic, and then you can use it. I don't think, Rudy, that we're really talking about here like a artificial iris or something, yeah, where they take 3,000-4,000 euro, or they handmade it. Yeah, that's a different story. I think that the EndoArt is kind of I wouldn't. You can produce it as a mass product if you want, without so much high cost. Yeah. So I don't see that issue. I understand what you mean, but I also see the cost that I have for my clinic for an eye bank, and also how long it took to get all the accreditation for everything, and so on.
But don't you think ophthalmology has done what ophthalmology often does? Because we could actually look into the eye, and we were the only clinical subset that could actually look inside their organ and have beautiful images. We didn't do much image analysis and image processing, and the brain people with CAT scans and computer complex algorithms sort of overtaken us in terms of imaging and imaging interpretation. And when you think about tissue preservation, damn it, every country has a sperm bank where sperm are, seriously, they're kept there for 20 years. And I don't think it's beyond the wit of man, if research is put into this. There's a lot of resistance from corneal surgeons who only want corneas from young people and only a few days old. But surely to goodness, these things are going to change. And in future, I think we will have more appropriate long-term storage, and that will bring price down and make it more acceptable.
Is that where the point where cell growth, regrowing corneal cells, 3D printing comes into play?
Yeah.
Well, it's a different area, but yes, you could bring that into play. But what I'm sort of saying is, when you think one of the most important biological techniques is to take a human sperm or human ova and many years later bring them together to a full-term fetus, etc. And, we've got a tissue which is mainly connective tissue with a few cells, damn it. And we know we can now plate endothelium and we can plate epithelium. So all we're really thinking about here is ways of preserving corneal stroma. So I honestly think there's a potential future here, and we ought to spend more time doing research in those areas. The orthopods are already there with long-term preservation of cartilage. The dentists are there with now osteoblasts from the buccal cavity. We've fallen behind again because we've had access to tissue.
Yeah, I can add to that. We have been working in private-public partnerships here in my province with the university, the hospital, the Technical University of Eindhoven, and DSM, which is a big company in also biomedicals. Probably John knows them. And I really think that the way to go will be stem cell derived tissue, and how we are going to do that. That's the question, of course. We've seen some interesting, well, Japan actually has made that, an area of excellent research, actually, but they put an enormous amount of money into it.
With the retinoids.
Yeah, also. And we actually, we have in the Netherlands here in Utrecht, in the lab of the Huybrecht Institute of Hans Clevers. I don't know if you know him.
Yes,
He was actually nominated, I think, on the side for the Nobel Prize this year. They have created an organoid from the tear gland.
Yeah.
So there's an organoid that can produce tears. I've seen it. I mean, they showed it. It was in the clinical research symposium of the ESRS this year.
Rudy,
it is fascinating. If you look at unmet needs, what we need, with these people with graft versus host disease, whatever you call it, this very severely ill people, Sjogren's, it's possible.
We already have a treatment in Harley Street for tears, and when some of my colleagues present the patient with bills, they start crying.
I think this is the best closing statement that we could reach. So I think the time is up. Anybody else to comment, or shall we close, Chris?
I have no more questions or comments. Rudy?
You know, I was asked to have a Sauvignon, so I'm going to, you know.
Ah, you brought your own wine. Excellent.
You said Sauvignon Blanc because Gerd had Sauvignon Blanc, so I brought one from Southern Tyrol.
Nice. Wait, John just ran out. Okay, John's back.
No, I'm back. It's just I was looking for a plaque. You know, for a period, I had an office in Harley Street, and my plaque said Professor John Marshall, and the plaque immediately beneath my plaque was the London Sperm Bank. And they gave me the brass plaque, but I can't find it. I'll show it next time.
Oh, good.
It's the reason why I'm so tired so often.
Right.
Okay, on that note, Eileen, Gerd, anybody else dare to offer anything after that? No, okay, we're good. We're good. All right.
Okay.
All right. Thanks everybody for joining. Hope our audience found this enlightening. Those of you who are tuning in later, you can address questions via the Oracle's LinkedIn page if you have any questions regarding sperm banks or other ways of managing human tissue. And on that note, thanks everyone, and good night.
Bye-bye.
Thank you. Bye-bye.
Good night.